This is Dr. Hutch, and this is a presentation on genetics and genomics. So genetics is the study of the inheritance of characteristics. Genes, which I'm sure you've heard of on a regular basis through the news and just talking about it regularly with people around you, are DNA sequences that encode specific proteins. And the genome is a complete set of the genetic instructions in your cells. It's written in the language of DNA molecules. It consists of a sequence of nucleotide building blocks, so A, G, C, and T, and that's adenine, guanine, cytosine, and thymine. All human genomes are actually greater than 99.9% alike in their DNA sequences. So it's just that 0.1% that makes the big differences you see around you. The exome consists of the protein encoding genes of the genome, and this accounts for less than 2%, or the 3.2 billion DNA bases of the human genome. Genetic information is encoded in DNA, and then we express it in RNA and proteins. And this influences your cells, your tissues, the whole person, and that is passed on to the next generation. The environment affects how genes are expressed and under what circumstances they're transcribed, and which messenger RNA will be translated into proteins. Cystic fibrosis is an example of a genetic disorder that results in defective chloride ion channels in cell membranes and results in thick mucus, which adversely affects a variety of organs, especially the lungs and the pancreas. Genetics carries power of prediction of traits in the offspring. And so knowing the gene distribution in meiosis and combinations in which they join at fertilization makes the calculations of probability that a certain trait will appear in the offspring of two specific parents possible. Patterns in which genes are transmitted in families are termed modes of inheritance. A karyotype is a chart that displays 23 chromosome pairs in size order. The normal karyotype shows 23 pairs of chromosomes. Pairs 1 to 22 are called autosomes, which determine traits, but not sex. Pair 23 is the sex chromosomes, so XX for female, XY for male, and this codes for sex. Each chromosome contains hundreds or thousands of genes. Human cells have 2 copies of each chromosome and 2 copies of each gene. Members of a gene pair are in the same location on homologous chromosomes. Alleles are various forms of a gene, which differ in DNA sequence. A person who has 2 identical alleles of a particular gene is going to be called homozygous for that gene. A person with 2 different alleles for the gene, heterozygous. This shows you what a normal human karyotype looks like. And in this case, notice that the chromosomes are really the individual chromatids at this point. They have not replicated into the second side of each chromosome like the typical chromosome you think of as like an X. Okay, the genotype is a particular combination of alleles for a particular gene or genes. A phenotype is the appearance of a trait or health condition that develops as a result of the ways the genes are expressed. The types of alleles, and often they only exist in 2 types, are dominant, recessive. In dominant, the alleles that mask expression of recessive alleles when they are present in a heterozygote. And recessive is an allele that may be masked by dominant genes when it's present in a heterozygote. In Mendelian inheritance, inheritance is explained where traits are regulated in dominant recessive manners. The phenotype of a heterozygote is determined by the dominant allele. A Punnett square is a table used to predict probabilities of genotypes in the offspring for certain traits. A square consists of 4 boxes. The mother's alleles are listed to the left of the boxes, and the father's alleles are listed above the boxes. Each box represents a possible combination of alleles at fertilization. A pedigree is a diagram showing family relationships and known genotypes and phenotypes for each family member. Circles represent females and squares represent males. The shaded areas represent affected people. Half-shaded areas are carriers, unshaded are unaffected people. An example of Mendelian inheritance, albinism. So an example of Mendelian inheritance is albinism, in which you have an autosomal recessive disorder. Albinism is caused by mutation in a gene that codes for an enzyme needed for production of melanin pigment. Melanin is responsible for color of the skin, hair, and eyes. Homozygous dominant, in this case 2 large A's, and heterozygous, which is a uppercase and lowercase a genotypes, have normal melanin and skin pigmentation. Homozygous recessive, with 2 lowercase a's, the genotype is albino, which means no melanin is produced. The possible genotypes for any offspring of 2 heterozygous parents is 25% homozygous dominant, 50% heterozygous, and 25% homozygous recessive. This figure shows you a cross between 2 heterozygous individuals. And notice when we put the mother on one side and the father on the other, and we split the 2 alleles apart here. So we have a large A, small a, and at the top, large A, small a, and that's the male at the top. And when we put them together, here are those results we just explained. Some other examples of Mendelian inheritance. In Huntington's disease, this is an autosomal dominant disorder in which a child will have the disorder if only 1 mutant allele is inherited. It involves loss of coordination, uncontrollable dance-like movements, behavioral changes, cognitive decline, and this begins in the 30s and 40s. The healthy or unaffected individual has a lowercase h and a lowercase d. And the one with Huntington disease would have the dominant. In cystic fibrosis, we have an autosomal recessive disorder. The abnormal chloride channels, thick, sticky mucus clogs the lungs and pancreas. For 2 heterozygous parents, offspring can have 3 different genotypes. Okay, we can have a homozygous dominant, and this is a healthy non-carrier. A heterozygous, this is an unaffected carrier. And then if it's homozygous recessive, the individual is indeed affected with this particular disease. Okay, this figure shows you the Punnett square and pedigree for Huntington disease. This shows you pedigree and Punnett square for cystic fibrosis. Okay, Mendelian extensions. Modes of inheritance which are not strictly controlled by 1 gene with 2 versions of the allele. Types of Mendelian extensions are codominance, incomplete dominance, multiple alleles, pleiotropy, and sex-linked inheritance. In codominance, inheritance occurs in which both alleles and a heterozygote are expressed. ABO blood type is an example of codominance. 2 of the 3 alleles determine the ABO blood groups are codominant, A and B. When A and B alleles are paired, they're both expressed. The uppercase A and B alleles are dominant over recessive O gene. In multiple alleles, we have inheritance in which there's more than 2 alleles. ABO blood type is also an example of multiple alleles, since it has 3 alleles, A and B, which are both dominant, and O, which is recessive. In ABO blood type inheritance, type A blood can have genotypes AA and AO. Type B blood can have genotypes BB and BO. Type AB blood only results from genotype AB, and type O blood only results from genotype OO. And incomplete dominance. This is inheritance in which the phenotype of a heterozygote is intermediate between those of the homozygotes. An example is sickle cell anemia. Normal hemoglobin allele, which is the capital S, does not completely mask the recessive allele lowercase s for abnormal hemoglobin in sickled red blood cells. Homozygous uppercase S genotype is normal. The lowercase homozygous condition genotype has sickle cell anemia. Heterozygous, which is uppercase S, lowercase s genotype, has sickle cell trait, and this is a mild form of sickle cell anemia. Another example, familial hypercholesterolemia. Increases blood cholesterol, raises the risk of developing cardiovascular disease. Homozygous dominant person has normal numbers of cholesterol receptors on the liver cells and can remove cholesterol from blood. Homozygous recessive person has no LDL receptors on the liver cells and may develop fat deposits around the eyes or joints and develops disease in childhood. Heterozygous person has half normal LDL receptors on the liver cells and develops disease in adulthood. Okay, this figure shows you incomplete dominance, familial hypercholesterolemia. Polygenic traits. These are traits determined by more than one gene. It's continuous variation, where you have a range of variation shown by polygenic traits. Examples are height, skin color, and eye color. In eye color determination, some genes control melanin synthesis, and others modify expression of those genes. In pleiotropy, a single gene contributes to several traits or shows multiple effects. People in the same family with the same genetic disorder may produce several different symptoms. Often it's seen in genetic diseases that affect a protein that's found in different areas of the body. An example is Marfan syndrome. This is an autosomal dominant defect. Defect in fibrillin, which is a protein found in elastic connective tissue in the lens of the eye and bones of the limbs and ribs and in the aorta. Possible symptoms are dislocation of the lens of the eye, long limbs, spindly fingers, caved-in chest, and weakness in the wall of the aorta. Different people with Marfan syndrome may display different symptoms. This figure shows you a polygenic trait with continuous variation. In this case, I like height. Okay, this also shows a polygenic trait of skin color. This is a 3-gene model. This figure shows you a polygenic trait of eye color, a 2-gene model. Punnett squares can also be used to track inheritance of whole chromosomes. In chromosomal inheritance, inheritance of a trait through the entire chromosomes exists. Sex is a trait inherited by inheritance of X and Y chromosomes. Females, XX, have 2 X chromosomes, and males, XY, have 1 X and 1 Y chromosome. All egg cells contain an X chromosome. Half of the sperm cells contain an X chromosome, and half contain a Y chromosome. Sex is determined by whether the fertilizing sperm is carrying an X or a Y chromosome. The probability of having a male or female child is 50%. Sex-linked traits. This is coded for by genes on a sex chromosome, and mainly X chromosome. The males carry only 1 X chromosome, so they exhibit recessive X-linked traits more frequently than females do. This figure shows you sex determination. Again, it's a basic Punnett square, and we put an XX here and an XY here, and then again we do the crosses and show the probabilities. And as you can see, chances of a female are 50%, chances of a male are 50%. Okay, X-linked traits. These are traits transmitted on the X chromosome. The X chromosome has over 1,500 genes, and they're much larger than the Y chromosome. Most genes on the X chromosome do not have corresponding alleles on the Y chromosome. Males express X-linked trait with 1 copy of recessive gene. There are no male carriers. Females with 1 recessive gene for X-linked traits are normal carriers and require 2 recessive genes to express the trait. Examples: red-green color blindness, hemophilia, Duchenne muscular dystrophy. In Y-linked traits, traits are transmitted on the Y chromosome. Y chromosome has only 231 protein encoding genes. It's shorter than the X. Some genes are unique only to the Y chromosome and are associated with male characteristics. This table shows you possible genotypes and phenotypes for red-green color blindness, which is an X-linked trait. Okay, sex determination. The Y chromosome. Maleness is derived from a Y chromosome gene called SRY, or sex-determining region of the Y. The SRY gene encodes a transcription factor. The SRY activates transcription of genes that direct the development of male structures in the embryo, while suppressing formation of the female structures. A female will develop in absence of SRY transcription factor. 3 groups of Y-linked genes are classified by similarity to X-linked genes. Genes at the tips of the Y chromosomes that have corresponding genes on the X chromosome for bone growth, hormone synthesis, energy metabolism. Genes that are similar to X chromosome genes are expressed in most tissues. Some are found only in males. And genes unique to Y chromosomes. These control male fertility and control of cell cycle, gene expression, and enzymes. SRY expression irregularity in non-binary sexes. In a reverse sex individual, if the SRY gene is expressed in the irregular manner, this can result in a reverse sex person. Results from the lack of suppression of development of female genitalia, and the person would have XY chromosomes and female genitalia. Crossing over of an SRY gene of the Y chromosome onto an X chromosome occurs with crossing over during meiosis. Chromosomal XX female is a phenotypic male. In intersex conditions, gene expression can also result in an incomplete sex reversal. The person can have aspects of male and female anatomy. This results from irregularities in the biochemical pathway that's started by the SRY transcription factor. Genetic determination of intersex. The SRY gene is responsible for the development of the testes and pathway for development of male reproductive structures. Genetic mutations in the pathway can result in intersex conditions. In congenital adrenal hyperplasia, cholesterol is converted by an enzyme into testosterone in males and females. A mutation in the gene that codes for this enzyme causes androgens, testosterone and dihydrotestosterone, or DHT, to accumulate in the blood. This can cause males to enter puberty at a very young age with enlarged penis. It can cause females to develop an enlarged clitoris that looks like small penis, facial hair, deep voice, although the internal reproductive organs are female. In genetic determination of the intersex, 5-alpha reductase deficiency. 5-alpha reductase is the enzyme that converts testosterone to DHT. Mutation of the gene that codes for this enzyme results in the deficiency of 5-alpha reductase and prevents formation of a penis in an XY person. It's identified as female at birth, but has male internal reproductive tracts. At puberty, the female characteristics do not appear. Male characteristics appear as adrenal gland produces testosterone. Penis development, facial hair, male musculature, and deeper voice occur. Androgen insensitivity syndrome. A gene on the X chromosome codes for androgen receptors. Mutation results in the lack of androgen receptors, so developing structures cannot respond to testosterone and DHT. This prevents development of male internal and external reproductive structures. XY persons with AIS appear female. Other factors that affect expression of genes: penetrance and expressivity. Expression of genotypes varies among people due to effects of other genes and environment. Distinctions of genotype expression: penetrance, all or none expression of a genotype in a person, and expressivity, the degrees of a phenotype. In complete penetrance, everyone who inherits disease-causing alleles expresses the phenotype. In incomplete penetrance, some people do not express the phenotype, even though they inherit the alleles. The majority of traits exhibit incomplete penetrance. Example, polydactyly is autosomal dominant trait. Some who inherit the allele have extra fingers or toes, and some do not. Variable expressivity. The manifestation of a phenotype varies in intensity in different people. Example, in polydactyly, people can have different numbers of extra digits or have extra digits on various extremities. Okay, genetic heterogeneity. Same phenotype resulting from the actions of different genes. This occurs when different genes encode enzymes that catalyze different steps in the same biochemical pathway. An example, hereditary hearing impairment. About 200 forms result from abnormalities in different genes, and each gene affects different aspects of hearing. Another example, blood clotting disorders. Several types of bleeding disorders exist and result from mutations in any of the genes that code for any of the enzymes in the clotting pathways. Mutations in different clotting enzymes will result in different clotting times. Hormone effects on phenotype. Hormones can affect structural or functional traits in different ways in males and females. Sex-limited trait. This affects the structure or function of the body that is present in only males or only females. Genes are either X-linked or autosomal. Examples, beard growth or size of breast. Sex-influenced inheritance. An allele is dominant in one sex and recessive in the other. Genes are either X-linked or autosomal. This is based on hormonal differences between sexes. Example, baldness allele is dominant in males, recessive in females. Heterozygous males are bald, but heterozygous females are not. Multifactorial complex traits. These are traits affected by factors inside the body or external environment. Examples, eye color, height, and skin color. Examples, common diseases such as heart disease, diabetes mellitus, hypertension, and cancers. Chromosomal disorders. So changes from the normal chromosome number of 46 produce syndromes due to excess or deficit of genes. Chromosomes can also rearrange as an inversion of part of a chromosome or exchange of parts between non-homologous chromosomes. Abnormalities in chromosome number may involve a single chromosome or entire sets of chromosomes. Rearrangements may interfere with a vital gene or result in gametes with incorrect amounts of genetic material. Aneuploidy. Euploid means the cells have a normal number of chromosomes. Aneuploid are cells missing a chromosome or having an extra one. This results from meiotic errors called non-disjunction that results in uneven separation and distribution of chromosomes. In non-disjunction, a chromosome pair fails to separate, either at first or second meiotic division, resulting in a sperm or oocyte that has 2 copies of a particular chromosome, or 0 copies, rather than normal 1 copy. When an abnormal gamete fuses with a normal gamete at fertilization, the zygote has either 47 or 45 chromosomes instead of 46. An extreme case of aneuploidy, polyploidy. This is the most drastic upset in chromosome number. It involves having an entire extra set or sets of chromosomes and results from formation of diploid rather than normal haploid gametes. If a diploid gamete participates in fertilization with a haploid gamete, the zygote is triploid, having 3 copies of each chromosome. Most triploid embryos or fetuses die, but occasionally an infant survives a few days but has multiple abnormalities. Skeletal muscle cells are multinucleated. They have more than typical number of chromosomes, but this has no harmful effects on health. Aneuploidy. Autosomal aneuploidy usually results in intellectual disability, since so many genes contribute to brain function. Missing genetic material is more dangerous than extra material. Trisomy. This is having an extra chromosome. And examples, Down syndrome or trisomy 21. This is the most common autosomal aneuploid condition. It's less severe than Patois or Edwards syndrome. In Patois syndrome, trisomy 13, usually results in a miscarriage. And if born baby has underdeveloped face, extra or fused fingers and toes, heart problems, or cleft lip or palate. Edwards syndrome, or trisomy 18. This usually results in miscarriage also. If born, the baby has similar problems as trisomy 13, unusual finger position, and extra flaps of skin in abdominal areas. In monosomy, this is missing a chromosome. Sex chromosome aneuploids. This is less severely affected than autosomal aneuploids. Turner syndrome as an XO often only delayed sexual development and infertility, but some have low set ears and extra skin on the back of the neck. In triplo X or XXX, this causes tall stature and menstrual irregularities, but not many other symptoms. Klinefelter syndrome or XXY. This causes sexual underdevelopment, breast growth, long arms and legs, and enlarged hands and feet. Jacobs syndrome, XYY. This causes a tall stature, tremors, and problems with language, coordination, and muscle strength and behavior. OY embryo does not survive due to a lack of genetic material from X chromosomes. This figure shows you how aneuploidy may occur. This table compares and contrasts trisomies 13, 18, and 21. Down syndrome. This is the most common autosomal aneuploid condition, and it can arise in two ways. In trisomy 21, you have 3 copies of chromosome 21. In translocation, you have 1 copy of chromosome 21 and exchanges parts with a different chromosome and a gamete and fertilized ovum receiving excess material from chromosome 21. Person has short stature, protruding tongue, thick lips, slanted eyes, abnormally shaped ears, heart or kidney defects, varied intellectual disability, increased chance of developing leukemia, and the person is slow to sit or walk. People with Down syndrome can learn to do some jobs, and some can read or use a computer. Chance of giving birth to a child with Down syndrome increases with age of the mother. At age 20, this occurs 1 in 1,667 births. At age 48, 1 in 14 births. Life expectancy has changed over the past 100 years, from 9 to 60 years of age. Prenatal tests. Several types of tests performed on pregnant women can identify anatomical or physiological features of fetuses that can indicate a chromosomal problem or defect the abnormal chromosomes. Ultrasound. This can detect eye fusion, cleft lip palate, extra fingers or toes, and nose malformation. Blood tests at week 15 can detect serum markers, chemicals that may indicate liver problems in the fetus. It's not very precise, so it's used as a screening test. Cell-free fetal DNA analysis. This is more precise than blood tests, so replacing it may detect trisomy. Chorionic villus sample. Obtained through cervix. Assume that these cells are identical genetically to fetal cells and can produce false positives or negatives. Amniocentesis. Fluid sample obtained from the needle inserted into the amniotic sac. Karyotypes from fibroblasts can show abnormal chromosome numbers or translocations. This table shows you the different prenatal tests. This figure shows you the methods of checking a fetus's chromosomes. This figure shows you an ultrasound exam of a 13-week fetus. Genomics in healthcare. Healthcare is starting to include clinical testing based on specific DNA sequences. Genetic tests can help with the following: detection of Mendelian or single gene diseases, identification of gene variants that indicate higher risk of particular conditions, selection of treatments that are least likely to cause serious adverse effects for certain patients. DNA tests can detect single nucleotide bases or entire genomics, or genomes. An exome consists of 1.5% of human genome, but includes 85% of mutations that influence health. In a patient with unusual symptoms, exome or genome sequencing may help with the diagnosis. Genome sequencing, along with family trait transmission, is important in interpretation of test results. In common conditions, genetic testing can help to refine a diagnosis and identify risk. An example, family inheritance of tendency for atherosclerosis, inappropriate blood clotting that could result in thrombus or embolism. Treating genetic diseases. Treatments either alleviate symptoms or involve targeting of specific genes or proteins. An example, cystic fibrosis treatments involve antibiotics to fight infections, drugs that break up sticky mucus, and taking pancreatic enzymes to digest food. Protein-based therapies correct faulty metabolic pathways by replacing missing enzymes or decreasing substrates. An example, Gaucher disease treatment, which involves replacing a missing enzyme, and it's very effective. Pharmacological chaperone therapy, this corrects misfolded proteins. An example, it's used in cystic fibrosis to refold abnormal chloride channel proteins. Gene therapy, this adds functional copies of genes to certain cell types or body parts. An example, it's used to treat hereditary blindness. And that's the end of the presentation on genetics and genomics.